they create a secondary immune response. So those are the ones that are produced by the memory cells. Okay? IgE has different functions, IgD and IgA. So different ones have different roles in the immune system, with IgG and IgM being the most prevalent in the immune response. Okay? So then you have the attack. So the antigen has been recognized. Now the antibodies are going to be released. Okay? The antibodies are going to bind the antigens, which causes neutralization, where the antibody binds the antigen, so the antigen cannot do any damage. Okay? Antigen will bind to IgM or IgG. This is the primary defense against foreign cells and bacteria. Agglutination, we've talked about that before. Agglutination can happen with any type of enemy cell, okay, where the antibody binds the antigens and causes the cells to clump together. This can happen with not just red blood cells. Okay? And then precipitation is a process that binds, a binding that creates these antigen-antibody complexes that precipitates these large complexes that can be phagocytized by macrophage. So this kind of gives you the process of humoral immunity. Okay? So step one, you have these B cells, and these B cells have these receptors on the membrane. Those receptors are actually antibodies, okay, that just happen to be attached to the B cell. They recognize the antigen, which is in green. It binds the receptor, okay, and then it's going to, B cell internalizes the antigen and displays the processed epitope. So it basically takes in the antigen, it processes it, and then you can see it presents the epitope, it presents the antigen to the surface. Okay? Helper T cells bind to the B cell and secretes interleukins. Interleukins are those chemicals that allow for communication. And then that's going to stimulate clonal selection, where interleukin stimulates the B cell to divide via mitosis. Remember mitosis, all cells are genetically identical. So you're basically replicating the cells, you're multiplying them. Then, once you have multiplication of the cells, then they're going to transform into different types of cells. The majority are going to become plasma cells. Plasma cells are antibody-producing cells. A small portion are going to become memory cells. And those memory cells are going to remember that antigen in the future. Okay? So for the exam, you don't have to memorize all these steps, but you have an appreciation for how it all works together. But I would never ask you an essay question to explain how humoral immunity works from antigen exposure to antibody production.
And that's why you're so cool.
Okay? So what you should have, what I do give you will be enough. It will be enough. So just kind of have an idea of just based on the study guide. Okay? And then the last step is memory. Okay? So you have those memory B cells. Okay? Remember, the first exposure is primary immunity. This is when you're first sick. Okay? That's why you're sick 3 to 6 days. It's the average time to be sick, roughly. That you have that 3 to 6 day delay. That's when you're going to have symptoms because your body is building up that immune response. You're creating those memory cells during that first period of illness. Okay? But when you're done being sick, you've defeated the pathogen, it leaves one with a memory of that antigen. Okay? Clonal selection, many cells become memory B cells, and these memory B cells, they're found in the lymph nodes. Okay? They're found in the lymph nodes, which are going to monitor the lymph, they cleanse the lymph, and they're going to bind, those antibodies are going to bind antigens as they pass through the lymph node. There's a quick secondary response where there's little to no effect, and this involves IgM and IgG. Okay? So IgM is that is what's secreted first, okay? More of a primary response. IgG is elevated for weeks to years. Those are the memory antibodies. Okay? But the memory of B cell memory is not as long as T cell memory. So this diagram here shows you the primary response. This is when you would get exposed first and you get sick. This is the secondary response. Okay? So you can see that IgM is produced first, then IgG. Okay? But you can see that there's a delay here. There's a couple day delay, 3 to 6 day delay in antibody production, because the body has to get the cells activated to produce the antibodies. Okay? So it takes a couple days for it to get jump started. Here, so then you get sick, you're creating the memory. Here you already have the memory cells, so the antibodies are produced from day one. They're produced from when you get exposed. And they're going to increase so fast that you don't get any noticeable symptoms. Okay. So that secondary response is much quicker because you already have the army formed. The defense, the army is already there, prepared to attack the enemy. Okay. So I want to point out that, I'll go through some of these. We have on Blackboard, if you look down here, I have several animations here that I'd like to walk through, just to kind of show you before we go. So this first animation here is of epitopes. And the epitope is a specific region, of the antigen that stimulates the response to the antibody. Okay?
Antigens are macromolecules, usually of molecular weight greater than 10,000, such as proteins and polysaccharides. They are recognized by the immune system as foreign. Individual antibodies are not made against the entire antigen molecule, but rather to particular chemical groups on the molecules known as antigenic determinants or epitopes. Many different antibodies can be made against a single antigen. Each antibody reacting with a different epitope. Complex structures, such as the surfaces of bacterial cells, may have many different epitopes. Each different antibody binds only to the correct epitope.
Okay. That gives you kind of a visualization of antigens and those specific regions. Now you have this one where I said where antigens are processed, okay? Where the antigen is taken up into the cell, and then it has to be processed and then presented on the surface of the cell.
Antigens are processed differently, depending on whether they originate within or outside the cell. Proteins produced within the cell, such as viruses or self-proteins, are broken down into fragments. Fragments of foreign proteins are antigens.
So this is the antigen.
The antigens are then transported into the rough endoplasmic reticulum. The antigens combine with class I MHC molecules. The combination is then transported to the Golgi apparatus and from there to the plasma membrane. Foreign antigens presented on class I MHC molecules stimulate cell destruction by activated T cells.
On the surface.
Self-antigens are not recognized by the T cells and do not stimulate cell destruction. A different process occurs if the antigen originates outside the cell. Phagocytes, such as macrophages, ingest foreign particles, such as viruses and bacteria, by endocytosis. The foreign particles are broken down into fragments within a vesicle. The vesicle containing the foreign fragments fuses with vesicles from the Golgi apparatus containing the class II MHCs, and the two structures combine. The MHC class II antigen complex is transported to the plasma membrane. The displayed MHC class II antigen complex can stimulate other immune system cells to respond to the antigen.
So the MHC protein presents the antigen to the surface of the cell. This one you can watch, it shows you the diversity of antibodies. This one I want to show, it shows you the activity of interferons. So we talked about what interferons are. And basically the cell is going to become infected with a virus, and it's going to show you how the interferons are going to warn the neighboring cells.
When a cell is infected by a virus, the virus enters the cell and produces structures that are not found in uninfected cells. The presence of this viral material signals the cell to produce interferon. The interferon moves out of the cell and attaches to receptors on nearby cells of the same type. The cell that produces the interferon is unable to save itself. The virus replicates in this cell and then moves out to infect nearby cells. The nearby cell that already has interferon bound to its surface responds in several ways, including production of enzymes that degrade messenger RNA and prevent protein synthesis.
These are
Thus, a virus can attach and enter the cell, but completion of the viral replication cycle is prevented.
So that first cell that got infected essentially sacrifices itself, but warns its neighbors. And now this last one is going to bring it all together, where it's going to summarize everything we talked about. T cells, B cells, and it's going to show you the complexity of how things interact.
Activation of the immune response typically begins when a pathogen enters the body. Macrophages that encounter the pathogen ingest, process, and display the antigen fragments on their cell surfaces. Macrophages with antigen fragments displayed on their surfaces are called antigen-presenting cells. An antigen-presenting macrophage interacts with a T helper cell that can recognize the same antigen. During the interaction, the macrophage releases a chemical alarm signal called interleukin-1, which stimulates the T helper cell to secrete interleukin-2.
See all those different chemicals?
Interleukin-2 causes the proliferation of certain cytotoxic T cells and B cells. The immune response from this point follows two paths, one using cytotoxic T cells and one using B cells. Normal cells of the body that become infected can also digest some of the pathogens and display antigen fragments on their cell surfaces. The body makes millions of different types of cytotoxic T cells. Each type is able to recognize a particular antigen. The cytotoxic T cells that are capable of recognizing the antigen displayed on the surfaces of infected cells bind to the infected cells and produce chemicals that kill the infected cell. Death of the infected cells results in destruction of the pathogen. B cells also come in millions of different types, each able to recognize a particular antigen. When B cells become activated by T helper cells, they differentiate into plasma cells. These plasma cells become antibody-producing factories, flooding the bloodstream with antibodies that can bind to the antigen involved in this infection. Antibodies bind to the antigens on the surfaces of the pathogens, marking them for destruction by macrophages. Some of the B cells do not turn into antibody factories, but instead become memory B cells that may survive for several decades. Because of these memory B cells, the secondary immune response to a future infection by the same pathogen is swifter and stronger. This powerful secondary immune response is what gives immunity to some diseases after you have had them once or after you have been vaccinated.
So it kind of brought everything together, concepts we talked about with antigen presentation, interleukins, interferons, kind of brought everything together to kind of bring the information that's on the PowerPoint slides kind of to life. So use these animations, use the videos to kind of help support the material that's presented in the slides. Okay? So next week, remember, make sure you do your connects. Make sure that you come prepared for the lab quiz. Study for your exam. Remember, tutoring is available. Okay. Also, we want us to remind you about, I put it at the top of the announcements, that to make sure you're filling out that COVID tracker, either using the app or the website. I would get into a habit of doing it every day, just not the days that you're here, to kind of be in a habit of doing it. Okay. So if you get symptoms, because I'm having students that have to quarantine. I'm having students that are positive. Make sure that you fill out that, you can contact me, but make sure you fill out that app, because that app information is going to go through a database. It's going to alert the college of your situation, and then the college will follow up with you. Okay? And that's it. Make sure you're filling out the app.
Girl, my feet had fell asleep. They are dead. I cannot feel my toes. Nope. Sitting in this chair got me.
Are you cold?
No, I'm sitting here. I can't sit.
It might be the chair because the blood going over.
It was terrible. They
Is there a
hurt.
sign-in sheet somewhere? Oh, right there.
Yep, just hanging out there. I am so happy. I have no reason to be this calm. Like, I wanted to ask you. Oh, go ahead.
Oh, I'm sorry.
No, go ahead. You're right there.
I submitted that case study with the lab, and that's why the lab said it was overdue. Is that an issue? Because that's why I thought you wanted was one submission. And it got-
So lab 3 should have been in its own folder and the case study in its own.
Yeah, and then, do you want me to resubmit it?
Sure.
Okay, that's not gonna cause an issue?
No.
Okay. All right, thank you.
Everything is cool.
Activation of the immune response typically begins when a pathogen enters the body. Macrophages.
This does look like an Oracle building, like a wood here. And I just think it's funny how certain things in the body have the same name, like Oracle, and then like what is the other one? Medulla. Oh, there's so much parallel. Did you ever hear about that lady? And I'm not going to keep you blabbing too long because that's why she's going to run away. About a lady whose organs were transferred, like her heart was on the other side, everything was on the other side. Did you ever hear about that lady? She didn't find out she was like 70.
There's a word for that.
Yeah, it's transverse. So I can't remember.
I forget the term.
Yeah, and it was
Everything that's on the right is on the left, and everything that's on the right is on the left.
right.
Flip-flop.
That's so crazy.
Genetics.
Yeah, that's cool.
Yeah, they making me go on 36th Street.
They don't have another one for you?
No, I live far.
Did you call them?
It's okay.
I would look on maps. I'm just like that though. I would be like,
Yeah.
hey, can I do it for this college? You know, I
Can
ended
you
up
do me a
having to go-
favor though? Can you send me
Yeah.
a picture of this? I don't have any storage on my phone.
Oh, you don't? No
No.
problem. I'll make sure you have a study guide.
Okay.
I'm putting together study guides for my 100 classes. You getting ready to sleep? Right now? You're not going, are you?
In like 30 minutes.
Okay, good. I just want to know because like I gotta wheel myself out, stretch my legs and all that. I'm just not ready to fly yet. Plus, I like your company. You're cool. Anything to keep me away from my children.
You was crazy.
Whew, really boy. It'd be like, Mom, can you go? No, I'm not. No, I'm not going anywhere. So yeah, I love the heart. Basically, what I'm thinking I'm gonna do, cause you know,
That's your study guide? Did you start it already?
I'm starting it right now. I'm starting my study guide right now. There's vocabulary, and then there's
This is
yeah.
what you found yesterday? When you
Yeah,
called
this
me?
is what I
Okay.
printed on
Yeah.
offline yesterday.
Yeah.
So, and then basically I'm putting together, you know,
Yeah.
what he gave us, and I'm taking a little peek at the connects, which I'm gonna finish up my connects tonight. So that way, I just hate, like this is what I was trying to do last night.
That's because I didn't submit my stuff yesterday.
Cool.
Yeah.
Oh, for
I only got
the,
2 assignments to do on math.
okay.
So I gotta do one more.
Yeah, you know what I did one time?
Oh,
Because you're using the Pearson's e-text. I actually found I don't know, accidentally, let me say, it's crazy. I wanted a YouTube video alarm clock, so I looked up one, because I just find her voice is so soothing. And after I had my little bleaching skin, I went to sleep in the last one because I was just fatigued. All right, so I'm just like, you know what? I need to start my day off with some math. And I slowed her voice down. I sped it up. I got through that, man. I'll help you if you need help. Because I like that damn
I
man.
just found the, so this is what I'm gonna do tonight. I'm gonna do what's due Monday tonight.
Oh, the case
'Cause I'm
study
already
on
up to date. I did all my reconnections.
on the senior lady. Do you have a copy of the
Print you out one?
Hmm?
Print you out one?
No,
This is for
I got it.
clinical.
I got it. I got it
Okay.
somewhere in the pile. But do you have the? What is that? It's with all the codes on it. I think I know. It's almost. I'm just tired, bro. I got like 3 minutes of sleep last night. Like 3 whole minutes.
Oh, my stomach hurts.
Salad?
Yep, how did you know?
I don't know. It had
I'm
had a lot of
lactose. I
cheese
told
on
you I'm
Yeah, it
lactose.
had a lot of cheese on it.
But I'm really, my mom and dad is too.
Is it lactose or is it a milk allergy?
Lactose, like it don't sit well
Okay.
at all. Anything dairy.
Ooh, girl, my feet numb.
So it's just the way your legs are. You sit in that chair. That chair's
Well,
too
yeah,
low.
when it's
Your
under
blood
here, it
flow.
just cut me.
It's your blood flow. The way you sit.
I know. Yeah.
Your legs are what?
No.
Numb.
You're cutting off nerves somehow.
Yeah, that says might be that chair.
They started getting.
Probably with the chairs, like.
How low it
the edge
is.
of the chairs.
Yep, yeah, it's cutting me right there.
Right there. Yeah. Cause yeah, I know how I feel. Cause when I'm in chairs that's too high and my feet dangle,
Yeah.
it's cutting me
Yeah,
off.
I got bad hips, I got osteoarthritis.
Mm-hmm.
So when I sit a certain way for too long,
Mm-hmm.
That's how I feel in there. Cause I have scoliosis, so
I get creepy.
Yeah, I feel like it's in my hip joint, like it's in my hip joint. I don't know. I'll tell you what, I miss my boo, but it's been a long year. I haven't had nobody to go on those long walks with. I'm stiff. Then I have fibromyalgia, so it's just a lot. But I don't know. I got a new bed. I got a Verlo. It's been sitting in the living room for a month and a half.
Your new bed.
Yeah. Because I had bought a bed from this place. It was like a store that I didn't know that the bed was made out of old and used materials. I did not feel comfortable with that, knowing that there'd be bugs.
Amazon has good beds too. I love Amazon.
Yeah, they had I forgot who had it, but they had a spend 1,000, get 500. So I went 500 with my grandma. She got a bunch of lamps and stuff. And then I got a bed. And a bunk bed. I threw out for my kids. So I gave them my bed. So they can have another place to just rump around in because they can't. It seems like my son-in-laws, when I get all 3 of my children together, they get rowdy. And then next thing I hear is, "Oh, Mom, no, no." And I go in there, and the furniture is dilapidated, things are broken, and nobody has any answers.
And then we got clinical. So this is the homework due for clinical. So we put this here. I'm gonna go home and do this. I just looked at my schedule and I thought I had to work today because I told you this my last time. They moved my schedule because remember last time I had to do second, so I had to leave early. So I work third shift now, and I'm bracing myself. I'm like, you know how you sit there and you talk to yourself like,
Oh yeah, oh no, this is what I gotta do type.